Human African trypanosomiasis, also known as sleeping sickness, is a current target of the World Health Organization, who lead a control and surveillance program to provide support to affected countries. The disease is prominent in sub-Saharan Africa, particularly in rural areas where access to healthcare is restricted. It is a parasitic disease transmitted to humans via the bite of the tsetse fly. Trypanosoma brucei gambiense causes 98% of infections, which occur in two stages. Firstly, the hemolymphatic stage, followed by the neurological stage, where the parasites have crossed the blood-brain barrier into the central nervous system. Without treatment, the disease is fatal. Accurate diagnostics are required, and currently diagnosis is carried out via a card-agglutination test to detect the presence of host antibodies. Professor Mike Ferguson and his team at the University of Dundee set out to improve on this test. Based on the World Health Organization's target product profile, they chose to develop a prototype lateral flow test, as it is cheap, stable, heat-resistant and does not require electricity. I met with Professor Ferguson to find out some more. First of all, his team carried out a series of tests to choose an antigen for use in the lateral flow test. They tested 4 soluble form surface variant glycoproteins, of which trypanosomes have hundreds. To their surprise, after testing by ELISA, one antigen in particular, SVSG117, showed remarkably high immunoreactivity with both stage 1 and stage 2 infected serum samples. Professor Ferguson explained that trypanosomes show antigenic variation. They can alter their surface proteins, and often a few have done this before the infected patient has completed development of antibodies against the first type. Once development is complete, all parasites other than those that have changed their proteins will be killed. SVSG117 is thought to appear early in infection, as all patients have the complementary antibodies to this antigen. Building on these results, 2 prototype lateral flow tests were developed. Firstly, a single antigen test to further analyse SVSG117, also a dual antigen test using native SVSG117 and recombinant ISG65, which Professor Ferguson and his team previously deemed to have useful diagnostic properties. A positive test was one that gave visual scores greater than or equal to 2 for ISG65 or 1 for SVSG117. Testing of the prototypes using samples from the WHO Human African Trypanosomiasis Specimen Biobank gave encouraging results. The sensitivity was comparable to that of the card-agglutination test, although specificity was lower. I asked Professor Ferguson what the possible reasons were for this. He explained firstly that the prototype was not optimised, and that people in these regions of Africa are often exposed to many infections and therefore have a high antibody content. These antibodies may cross-react with the antigens on the testing pad, resulting in the false positive result. However, optimisation of the test can improve upon this, and this can be done, for example, by altering the chase buffer. The work of Professor Ferguson's lab has been fundamental, and in fact, in September of this year, a rapid diagnostic test has been released based on their research in collaboration with a group at the University of Cambridge. The test uses recombinant ISG65 and a different soluble form variant surface glycoprotein. The antigen used is a recombinant protein rather than a native one, which is necessary to further meet WHO criteria by removing the need to cultivate antigens in rodents. The test has been officially launched as part of efforts to eliminate human African trypanosomiasis by 2020.